Nikki, thanks so much for the invitation um, to talk. I'm really excited to share my work with this group, and thanks also to Zach Sieb, who I think is no longer with the IPM Institute, but for the initial invitation to share my work. My name is David Allen. I'm an associate professor of biology at Middlebury College in Middlebury, Vermont, and I'm going to talk about using tick population dynamics models to assess control strategies. So just a brief outline of what I'm going to talk about today, this afternoon, is just talk broadly about models in public health. And this will be from a little bit of a wider frame so that I can give some examples of models that have been used to guide public health interventions in some sort of relatively high profile and large impact examples beyond the world of ticks. And then I'll focus in on modeling tick control strategies and look at some recent literature on folks who've tried to model tick control strategies to get some insights about the best way to do that, to the best application of tick control strategies, and then get into my research and have some conclusions. Okay, so why would we use models to guide interventions in public health? So we can use them as virtual laboratories. We can test strategies that would be too expensive, logistically challenging, or even unethical to test in the real world. And so then we can do some scenario analysis. We could say, what if we were able to vaccinate 50% of the population or 80% of the population? How much would we reduce disease transmission? Would we get that R naught down below 1? And so would we expect the infection to die out? We can also use these models to optimize limited budgets. So we can say we have this range of different potential interventions, like we could put out 4 posters, we could put out bait boxes, we could spray with caricide. Which of these is going to give us the most cost-effective intervention in terms of reducing number of ticks per dollar spent? And models allow us to test these different scenarios. So like I said, I'm going to start with 2 models that are outside, or 2 examples that are outside of the tick frame, or to give some examples of places where models actually have been used to guide, again, like large scale public health interventions. So the first one is rubella. Rubella is caused by a virus, results in, if contracted during childhood, a mild, sometimes even asymptomatic rash. But if it's contracted during pregnancy, then can result in a very serious congenital rubella syndrome, which leads to deafness, blindness, can lead to deafness, blindness, and heart defects in the newborn child. So a vaccine was developed, and then models were used to say, okay, what's going to happen as this vaccine is initially released? And one of the interesting results that was found was that partial vaccination could actually make the situation worse. So in the pre-vaccination setting, or no vaccination setting, the virus circulates freely. And this is sort of like, you could think about it like chicken pox before there was a vaccine for that, where most individuals get infected relatively early in life, in childhood, leading to this lifelong immunity and progressing into childbearing years. And so most folks contracted it as this mild childhood illness, and then didn't have a risk of it during pregnancy. The worry was then, in a low coverage setting, that the would slow transmission of the virus, but not eliminate it altogether. So then there's just less transmission of the virus. So some people miss that childhood infection because it's just not circulating in as much. And so people who both miss the childhood infection and don't get vaccinated because of the low coverage become susceptible in adulthood. And then there's the chance that they get infection in this childbearing years, and it would ultimately rise the level of congenital rubella syndrome, even if the overall rate of rubella is lower. So we'll see a result from this modeling. This is a paper from the 80s that did it. I've sort of recreated the figure just to make it a little clearer for the graph. Um, but what we have is the year that the vaccine is released on the x-axis, and then different um, levels of uptake of the virus of the vaccine. So this is an example of one of the real advantages of modeling is we can, quickly say like we have these different scenarios of how much vaccine uptake there is, and then we can play it out over 20 years. So something that you couldn't do experimentally. The dotted line on the y-axis is the sort of like baseline level of CRS um, right at the time when the vaccine is released, or if they didn't have any vaccine. And initially, because the vaccine reduces the level of rubella that's circulating, there is this reduction in CRS incidence. But then this also means that in these cases with low levels of vaccine uptake, these greener and yellower colors, um, there's people who miss their childhood infection, and so then once they enter their childbearing years, you get this increase in the level of CRS down the line. And it's not until you have relatively high levels of vaccination above 80% that you ultimately reduce the amount of CRS altogether. So this was the model, but in this case, the insights from the model were put into practice. So in the United Kingdom, they rejected universal toddler rubella vaccination. They had recently released a measles vaccine, and they could see, the level of vaccination uptake that they were getting with this measles vaccination was relatively low. So they didn't think they could also get it high enough for rubella. And they just chose this selective vaccination of schoolgirls above a certain age to allow the virus to circulate freely among younger children to build up that natural immunity. They didn't then ultimately choose to pursue universal vaccination until they had the MMR vaccine, measles, mumps, rubella. They'd built up the infrastructure to ensure that they would, or to hopefully get above 90% vaccine uptake. The counterexample here is Greece, which did introduce universal vaccination, but the coverage stagnated. The average age of infection shifted to from childhood to young adulthood, just as predicted in the model. And Greece in the 90s suffered an outbreak of CRS, again, like the model predicted, that 20 years after the release of the vaccine, you might ultimately see this increase in CRS if the vaccination level is low. So one example where a model was used to, again, give these predictions of different scenarios, what's going to happen over long term into the future, and that guided public health intervention. A second example, this one also from the United Kingdom, was the outbreak of foot and mouth disease in 2001. So foot and mouth disease is this relatively serious and quite contagious virus of even-toed ungulates, cattle, sheep, pigs. And there was an outbreak of livestock in the United Kingdom, and the government followed their standard protocol, which was to cull animals on farms with confirmed infections. The real-time data was coming in. It looked like the epidemic was still spreading. And so they fed the real-time data into models that showed with this current strategy of only culling on infected farms, the epidemic would continue to spread and infect thousands of farms. So these models said, you need to sort of get ahead of the virus. This intervention is currently not getting ahead of the virus, not controlling the outbreak. So they tested multiple interventions in these models that were models of the spread of foot and mouth disease across farms in the UK. And they came up with two potential interventions that were more aggressive, and they thought, based on the models, would get that, R naught down below 1. So one was this contiguous cull policy where animals would be culled on infected farms within 48 hours, but then on all neighboring farms that shared a border within 48 hours. And so this is where you get this contiguous. Or a policy where you would cull even larger range around infected farms within 3 kilometers to create this even additional fire break of the virus. Ultimately, the UK government adopted these policies based on, again, on the insights of these models, applied the contiguous cull policy nationally, and the 3 kilometer cull in the worst hit regions. Over 6 million animals were culled, massive logistical effort that ultimately did halt the disease. So this is a very costly and had, obviously many animals had to be killed, many of them healthy. And so afterwards, they wanted to look back at this strategy. So Ian Anderson was appointed on this inquiry, and they largely endorsed the modeling approach and the contiguous cull policy. And retrospective modeling demonstrated that maybe it definitely shortened the epidemic and maybe ultimately reduced the number of animals that had to be culled, because if the disease had spread even further, more farms would have been infected. And the epidemic was effectively over in September 2001. There was a debate, and some people disagree with these findings that maybe the models they had unvalidated assumptions, overestimated the spread, resulted in unnecessary culling. Critics also said that some of the epidemic may have peaked before these policies were implemented, and credited national bans on animal movement. But this is just another way to highlight that models have been used to guide large-scale interventions in public health. Okay, so now we'll look at modeling for tick control strategies, where the approaches are newer, but is increasingly been applied of tick population models that have then been used to test tick control strategies. So I'm going to highlight 2 recent papers from the literature just to show, again, the range of possibilities of how this can be used and some of the insights that have been found. So I'll talk about 2 here, Carrera Pinheiro, vaccination cost effectiveness. So I like this paper because they do this cost effectiveness analysis for a rodent targeted vaccine. And then Ruiz Carrasco, who tested multiple possible tick control strategies to see which was the most effective. So this first one, this vaccination cost analysis, it was based on this mouse or rodent targeted vaccine. So bait that has a Borrelia vaccine in it. The mice eat the bait, and then they become they're vaccinated against Borrelia, and so that's going to slow the spread of the Borrelia in the enzootic cycle between mice, these important reservoirs, and ticks. And then what they did in this case was they thought, like, is this intervention cost effective? If you think about the cost of deploying it compared to the cost of avoided cases of Lyme disease. And they used a model, this system of differential equations that modeled the enzootic cycle between ticks and mice, and then also contact rate between ticks and people. So in this case, they need to have some parameters to put into the model to see whether this, again, is cost effective. So they have a cost of like how much like per unit increase in vaccination effort cost. So this is deploying this rodent targeted vaccine. They have a cost for the average cost of Lyme disease treatment. So this was from the literature based on electronic medical records, compiling this estimate of the nationwide estimate of Lyme disease costs per year, but in this case, just per case. And then these parameters that they varied based on the contact, so or the context. So a contact rate between mice and vaccine. So this is as they increase their vaccination effort, how much are mice picking up these vaccines to sort of find this estimate of like what's the optimal amount of vaccine in a setting. again, based on cost effectiveness, the biting rate of ticks, so how many of these ticks are out there biting humans. And then they also have an idea of the context in terms of how many susceptible humans are in that setting. So is this a suburban park or a trail or a neighborhood? So I'm just going to give a couple of their results again because I think it's an interesting application of using these models to ask real world questions, but not yeah, just to highlight some of these results. So again, I've modified the figures from, or adapted the figures from the paper just so that they're easier to read here on the slides. But here on the x-axis is this vaccination effort. So this is a measure of like how much of the vaccine they're putting out there. And then on the y-axis is the cost, the dollars saved. So the cost of putting the vaccine out there versus the cost of avoided, or the benefit of avoided Lyme disease cases. So in this high traffic trail, if not very many ticks are biting, then there's no amount of vaccination that saves money. So, if you're right at zero, you're not spending any money, you're not saving any money. But as soon as you add more vaccination, the intervention just becomes costly. But as they increase the biting rate in their model, they can find that low levels of vaccination are cost effective. And so at the highest biting rate, then they do find that this certain level of application of the vaccine it results in savings ultimately to the if you compare the cost of putting out the vaccine versus the cost of Lyme disease cases. And in a suburban park, you have even larger savings here, this because they're modeling in this case that there's more humans around. So more likely that, if these ticks are out biting, they're going to bite a human. And over a wider range of parameters. And yeah, they find cost savings and then ultimately a higher optimal rate of vaccination effort for saving money. Second example is from Ruiz Carrascal working in the Duke-Wasser lab, where they have a really nice tick population model, and then they can put into their model these three different possible interventions, either reducing deer to reduce the tick population that way, applying this metarhizium bio pesticide, fungal biocontrol, or putting out bait boxes where rodents will come into the boxes and then they're treated with fipronil, this acaricide. So they can, again, an advantage of the models is they can, say like, what's the effectiveness of just doing one strategy? What's the effectiveness of combining multiple strategies or even all three strategies? And then they're going to say, what if we put out this strategy just for 1 year, or what if we do it for 2, 3, or even 4 years? So here's just the first result where you've got deer, metarhizium, and fipronil. And what they find is that these bait boxes are the most effective at reducing the number of ticks. It reduces the number of ticks by 50%. Combining the bait boxes with deer reduction or metarhizium or all three, you don't get that much more added reduction because you're already about at 50%. If you continue these interventions for multiple years, then the results start to compound. But the other big thing you see is that the deer reduction here then sort of starts to really pay off in year 2. And this makes sense given the tick life cycle, that what they're looking at is a reduction in the number of nymphs as that main life stage that is transmitting Lyme disease to people. And because of the time lags in the tick life cycle, by reducing deer, you don't see a reduction in the number of nymphs until a couple of years out. And at that point, the deer reduction becomes even more get more of a reduction than the bait boxes, but they're about equal. And combining, again, you don't get that much additional reduction because the deer or the bait boxes on their own are quite effective. In this case, they don't find the metarhizium very effective. They parameterized this model based on their own tests. So they put out they did each of these three interventions, and then they measured how much of an effect they had, and for them the metarhizium just wasn't that effective. Okay, so these results make you think like, oh wow, these are really, really exciting. You can put these different interventions into a model. You get these really precise outputs. What's the catch? And the catch is that, as probably you're well aware, models are inherently limited tools. And I like this quote from the statistician George Box that all models are wrong, but some are useful. So what does this mean that all models are wrong? It means that all models are simplifications of the system they represent. So the world is very complex. Tick life cycle, all of its hosts, its interaction with the abiotic environment, with the climate, is very, very complex. And there's no way we could encapsulate everything that is driving tick populations into a model. At the same time, some models are still useful. If we are able to distill down the salient factors that drive tick populations, encode them in a realistic way into the model may be able to do a good enough job of telling us how many ticks we expect in a given setting, so that even though the model is wrong, it's not 100% giving us a representation of tick biology. It's still going to be good enough that it can be useful. So what we need to avoid is that the simplifications that we introduce into the model introduce unrealistic assumptions. Because if the unrealistic assumptions are unrealistic, then the model outputs, again, will be unreliable. If the model, doesn't actually reflect the biology of ticks, you put something in, you can't trust what comes out. So just to highlight that, I'm going to return to this vaccine cost effectiveness paper to just think about one of the assumptions they put into the model. They treated white-footed mice as the only juvenile host and the Borrelia reservoir. So this is done in some models where you effectively have two hosts. You have like a deer host that feeds the adults, and you have a rodent or mouse host that feeds the juveniles. Um, and so it's a simplified host community structure. And in that case, if you then vaccinate the mice, you're going to be clearing the juvenile host that's that's the Borrelia reservoir, and that's going to solve the problem. The issue here is that this is going to ignore other important juvenile tick hosts and Borrelia reservoirs. So there's this rich community that the black-legged tick is feeding on, including shrews and chipmunks that are also, again, important tick hosts for juvenile ticks and can be very, very effective Borrelia reservoirs. So if this model is going to, especially shrews, are going to ignore, a bait that's designed for mice because they're insectivorous and not omnivorous, like white-footed mice, they're not going to be interested in that bait. So they're not going to be picking up, in this case, this Borrelia vaccine. This model's going to miss that. And so it could overpredict the effectiveness of these mouse-targeted vaccines if you have a lot of shrews in the area. So this isn't to like single out Carrera Pinheiro. I think this was a really cool paper. I love the way they do this cost effectiveness analysis, but just to highlight that, all these models are going to have limitations, and we have to be aware of them when we interpret the output. Okay, so that is transition to the research that I'm doing. So like in these previous settings, I'm going to start with a tick population dynamics model. And like I highlighted, modeling is a tricky thing. We can't totally capture what's happening in nature, but we want to be able to put enough complexity into the model that we think it's capturing the salient features of a system. So in this case, I break up the black-legged tick population into these life stages where I think, we have to just break it up into these discrete stages that have their own unique biology. So when a tick is an egg, it's different than in a closed larva. And then after that, a closed larva goes through a hardening period, then it's active, questing, feeding, engorged. So in this case, the model tracks the number of ticks in each of these life stages. Then transitions between the life stages reflect this, life history transitions of either an egg hatching or a questing larva going to feeding when it finds a host. And these transitions are again driven by what we think are the salient features of what's driving tick populations. So in this case, that's the abiotic conditions like temperature and vapor pressure deficit that determine, the temperature determines how fast a tick develops, or vapor pressure deficit measure of the drying power of the air is going to determine how active ticks are and their desiccation rate or dying rate due to desiccation. So that's one thing that determines the tick population. And the other is the host community. So how many of these important juvenile or adult hosts are out there that will determine how likely it is a questing larva finds a host and becomes feeding. Or if a larva is feeding, how permissive is that tick host so that then it becomes an engorged larva that successfully can molt to the next life stage. Um, so Here is the model in my case is driven by these let me just um, driven by these predictors, which is the temperature and the vapor pressure deficit, which determine um, again, tick development rate, tick activity, and then the host population. So the number of hosts that are out there is going to determine, how successful ticks are at finding hosts and then molting to the next life stage, or how successful an adult host is, a female adult, at finding a host and then being able to lay eggs. And we want to have a host community that balances simplicity with realism. So there's no way that we could model, the wide, huge range of species that ticks possibly feed as these, there are these extreme generalists, black-legged ticks. But we want to include the species that are feeding a large number of larval, nymphal, and adult ticks. So the host community that I chose in this case to reflect the host community in the northeastern United States is Peromyscus mice, so deer mice, shrews, which are these important, um, uh, important hosts of juvenile ticks and reservoirs of Borrelia, chipmunks, squirrels, and deer. The model is written in R with an R package that I've written with a student. And the goal of this R package is to make it easier to write, run, and analyze tick population dynamics models. So we want to make tick population modeling more accessible to a wider audience. So we wrote this package. That allows our user to really quickly download a package and specify a model relatively simply. And then in the background, the model does the running and will make it easier to analyze the output. So you take up the model's going to assume you have some, or you need to have some life cycle. So this is just a more simplified life cycle to sort of illustrate it, and then transitions between life cycles, and you specify them as, in this case, maybe the transition from egg to larva is a function of temperature. So how fast that development is depends on what the temperature is. Um, this model or this package is up on our CRAN, the R package repository, has a GitHub page where you can, see some example usage and some example models, and then a paper in Parasites and Vectors where we describe how the package works. But yeah, to return to this model, so we wrote this model, and then we have this question of, okay, we have a model, we know it's wrong, it's a simplification of a very complex biological system, but is it useful? Can we do at least a good enough job of predicting how many ticks are out there so that we can trust the output? And that's the process of model validation, where you take your model and then you compare its predictions to real world data. The model validation was done at my field sites in Vermont. So Middlebury is here in Western Vermont, and I have a set of field sites in this area where we use an elevation gradient in the Green Mountains to get sites of different climates so that we can then input those different settings into the model to see if we can predict the number of ticks. So on the left side of this map in the western side is the Champlain Valley, low elevation along, the Champlain Valley by Lake Champlain, and then the right or east side is going up the Green Mountains higher in elevation. So in addition to the number, so we've been sampling for ticks over the last decade, and also getting a sense of the host community with Sherman traps and camera traps, and tracking the climate or the temperature and vapor pressure deficit. So the model validation going to look like this. So on the x-axis, we have the model predicted number of ticks, and I'm using nymphal density just again because it's this key life stage for transmission to people. And then on the y-axis, we have the observed number of ticks. So that dotted line down the middle is, if the model was perfect, then all the points would fall out right along that line. And we want to be as close to the line as possible to say that the predicted and observed are similar. And because elevation, I know, is one of the key drivers in this case of giving different numbers of ticks in my sites, I'm just color coding elevation. So this is what we find, and although the line doesn't fall, exactly on that dotted line, it's going in the right direction, and it is relatively close to the dotted line. It does much statistically better than just random. At the high elevation, these sites again with the yellow greens, those are all right at close to the origin of the graph. So the model predicts they won't be very many ticks. We don't find very many ticks. Versus the deepest blue color, the lowest elevation sites, those are all in the upper right. The model predicts that at these warmer settings, we should find more ticks, and we really do find more ticks. So the model does a good job if it explains a significant fraction of the variation in the number of ticks. And so in this case, we're going to call this model validation. We're going to say that the model can do a relatively good job of predicting how many ticks are out there, although not perfect. Another aspect of model validation in this case is tick phenology, when ticks are active. So if we want to predict, when should we put our intervention out there, the model has to be able to accurately predict when the different life stages are active. So this is going to be another way that I want to try to validate the model. So again, low elevation versus high elevation for my sites, there's big differences in the number of ticks, or when the ticks are active in addition to the number. So here you see the season, and when ticks at low elevation are active, larvae at low elevation are active. So most of these larvae at low elevation are active in the late summer, early fall. So these are larvae that hatch that year, and then they're active in the year they hatch. But a small number either don't find hosts and overwinter, or they don't hatch quite in time, and you don't see them until the following early summer. So that peak in June is actually ticks that hatched the previous summer. And the model can reproduce this bimodal period of activity. Doesn't get the exact date right of when that peak should be, and the questing period of the larvae in the model is a little bit more sort of like spread out, not as concentrated. High elevation, we don't find late summer ticks at high elevation here. I think it's colder. The ticks aren't able to quest in the year that they hatch, and so you don't find the larvae until the following early summer. The model also predicts this. You only have this one single peak of early summer larval activity. It does a relatively good job of lining up when that peak should be, but again, it sort of predicts that the ticks will be active for a much longer window than they actually are. So some things still to try to fix with the model. Um, it does a quite a good job of predicting the activity of nymphs at both low and high elevation. Predicts right about when it should be, although it misses like a little bit of a late season bump in nymphs at low elevation that sometimes we see. Okay, so we're going to consider that, that the model is able to reproduce many of the salient features that are out there. And so now we can say if, we trust that output of this model, we can now use it to test different tick control strategies, not unlike what Ruiz-Carrascal did. So I'm going to try two in this case. I'm going to use test a metarhizium, like fungal biopesticide application, and then these fipronil-based bait boxes. And I am not, I didn't then like test how these work, the direct effect. So I'm going to use a literature review to then say, okay, if we do spray with this metarhizium, what's the change in some of these life stage parameters? So in this case, spraying metarhizium, going to the literature, I can see how much that increases larval and nymphal daily off-host mortality. And then I can encode that into the model. And then same thing for adult daily off-host mortality. Then the other thing that I need is some sort of like measure of how long the metarhizium in this case stays active. So I'm going to code that into the model as sort of like a half-life. So how long before the effectiveness of this intervention is cut in half as the spores lose their viability or wash out of the system. And in this case, this is about a month. Same thing for these bait boxes. Here's a nice picture from a paper by Erica Marchtinger, where she put up a camera over the bait box. You see this little white-footed mouse going into the bait box. So they go in there to get the bait, and then they get fipronil that should treat the ticks on the mouse. So again, did a literature review to find if a mouse does go into one of these bait boxes, what's the rate of larval on-host mortality? What's the rate of nymphal on-host mortality? So I can then encode those into the model. And then also, what's the intervention half-life? So if you did just put the bait box out and you didn't refill the bait or reapply fipronil, how long before the effectiveness has been cut in half? And in this case, that's about 2 months as the bait's eaten, the fipronil loses its effectiveness, or a raccoon damages the box. I'm going to follow the same sort of rough scheme for these graphs just so we can compare them more directly with Ruiz-Carrascal. But interestingly, I find the opposite of what they do. So I found that the metarhizium is relatively effective and these bait boxes are not very effective. And then when you combine the two, you don't get much additional because the bait boxes weren't very effectiveness when I tested them in this model. And continuing that out for multiple years, you get these compounding results until the end where this spray over those 4 years really has relatively big effects, but the bait boxes have a muted effect. So just to compare, that's the opposite, unfortunately, from the Ruiz-Carrascal, where they found that bait boxes are of a very big effect and the metarhizium very not much effect. So this highlights that these results can be idiosyncratic based on the models. For metarhizium, I think the reason here is just, their measurement of how effective it was in their field trials was relatively low. And so then they encoded that into the model and it didn't have a big effect versus the literature review that I did showed a little bit more effectiveness. So for the bait boxes, I think the difference here is sensitivity to ecological context. So the host community that I've used here is, I've got white-footed mice, I've got shrews, I've got chipmunks, squirrels, and deer. And the first four of these are going to be important juvenile hosts. but only mice and chipmunks will visit the bait boxes, and shrews aren't going to visit them because they're insectivorous, and then the squirrels are too large. So this is a way that we can again use the model to see like how sensitive is the model to this ecological context. So if we then, ran the model with this low diversity where we just upped the number of mice and chipmunks, but we don't include any shrews or squirrels, then the effectiveness of the bait boxes really does increase. And then, in this low diversity setting, the bait boxes now start to have much bigger effects because the remaining juvenile tick hosts are actually visiting those bait boxes. So I think what's happening is that, at least in my model with my host community, there's all these other hosts that are out there that are not visiting the bait boxes, and that dampens its effectiveness. One more, application of how you could use these models is like, okay, maybe in this case, the metarhizium is the most effective intervention. We could say, well, then what's the best time to spray? In the previous one, I just arbitrarily chose June 1st as the date of applying the intervention, but we could go back in and say, okay, what's the most reduction in the number of questing nymphs if we sprayed on, the first of every month? And then we can test, very quickly in the model what the result is. And in this case, again, just given the particular phenology at the site that we're in, you get the best reduction in ticks if you spray on May 1st. And that makes sense just given, that's sort of the beginning of the nymphal season. And so if the metarhizium has this half-life and that like would then take it through this, most important time of activity. Okay, so just some quick conclusions. Models provide these powerful tools to test tick control strategies, but you really have to be validated first so that you have to say like, that we can only trust the output of this model if it's actually been validated against independent data where it can actually predict tick density. Even with this validation, it's important to understand sort of like the limitations inherent into your model. Maybe it was validated against one particular set of data. Maybe it couldn't be applied outside of a very different setting. Um, and still, though, models are these powerful tools to compare the effects of different tick control strategies, see how those strategies depend on context, like, how does it depend on if we did it in a, high human activity setting versus low human activity setting, this host community versus that host community, test specifics of applications, like how much of the vaccine do you put out, or when's the best time to spray, and then do these cost-benefit analyses as well. So thanks so much to Zach and Nikki for the invitation to present to the IPM Tick Working Group. Thanks a bunch to some Middlebury College students who helped with this modeling effort, and then countless Middlebury students who have contributed to the tick sampling dataset over the last decade, and funding from a number of sources. Happy to take some questions.
Thank you so much, Dr. Allen. Cool stuff. Yeah, we can take some questions and I'm gonna launch the post-webinar question too. And people can put their question in the chat or raise their hand. Go ahead.
Thank you so much. That was really interesting. And I'm kind of hoping for a plug-in information and play, and it sounds like your package might be that. But I just want to check my assumptions. It looks like this is really geared towards Ixodes scapularis, and I'm thinking it will not work for the other species. Is that correct?
Yes, yeah, unfortunately. Yes, yes. It the specifics of, all of these life stage parameters that are I guess I should have said that taken from the literature and specific to Ixodes scapularis. Um, I think that there has been some modeling for some of the other ones, but yeah. I mean, I think you could maybe squint and use some of the outputs, but it probably would be better to do it for a specific one for each species. In terms of, it's not quite plug and play. I think I tried with my student to make it as user friendly as possible, and I think we're still tweaking it, but I'm happy to help folks if they want to try it out or share some of my code.
And if I could have a follow-up question. So I'm in the Capital District of New York, so I'm not that far from you. So I would think these models would probably do well where I am.
Mhm.
I spent the morning talking to school grounds managers on Long Island, which is a very different climate, community,
Mhm.
soils. So also, do you think that those parameters are going to follow through across those kinds of differences?
Yeah, I think my hope would be that the model could apply within like a broad ecoregion so that it could apply across the Northeast, maybe. And then but the exact line of like where then you get down to the mid-Atlantic and it no longer applies or the Midwest. Um, but you can sort of modify what the host community is, and so you could put in different amounts of the different hosts, and but I think, yeah, with local adaptation and differences in like you said, in soils, I'm not exactly sure where the line is, but my hope would be broadly the Northeast. Yeah.
All right, thank you.
I'm going to read this question. I was wondering if you could speak at all to your process on standardizing field collection data, say how to quantify overall tick abundance if you have more people sampling one year and fewer another, or visit a certain field site more times.
Yeah. So yeah, we try to be very standard with our sampling where we throughout the field season, we visit each site every other week. But you're right, I do have a wide range of students, and it does seem that some of them are better at seeing the ticks on the drag cloth than others. We try to standardize across that. But in order to build this long-term data set, um, I have, I have a methodology for going out to each of the sites every other week during the tick season, and have a set plots that we drag. We roughly follow like the neon sampling method of, yeah, yeah, Thomas.
I may have missed what you said, but because of the significance in your model of the hosts, it looked like you had shrews at a very high importance relative to mice and chipmunks. And how many sources did you use to derive that number?
Yeah, this is a really good point. So the mice numbers are from my own trapping and then mark recapture. So I guess the shrew data is, yeah, it's harder to track shrews and to get their population density, and so this is from a literature review, but it does come heavily from Osfeld data set, which, where they have estimates of the number of shrews, and so the shrews are quite inconspicuous, but when folks have tried to sample for them specifically, they often find many of them. So this is 50 per hectare compared to, like 35 mice per hectare. And so, yeah, I think you're right. If the shrews are not actually that common, then that would change the outputs of the model.
So is this 50 and 35 is individuals per hectare, I guess, is what you're saying. So it's assuming that shrews are a third more abundant in the habitat than mice.
Yeah, that's right. Yeah.
Mhm. I'm a little skeptical about that for sure.
Okay. Yeah.
And that influences pretty strongly, I think, your outcome. Did you also have ticks per each of these species of animals figured into it? I might have missed that too.
Yeah, that is figured into it. So there is a there's a tick. When questing ticks are going to attach on to the hosts based on a calculation, which is if there's a certain number of ticks out there, and then you find that many, the body burden, it sort of back calculates out the rate at which each a tick attaches to each host.
So
So
you didn't have a validated study that you were either following in terms of the burden of ticks on each of these types of animals.
yeah, no, that so I started with studies and across a bunch of many different studies, average body burden at peak tick questing, and then based on that body burden, um, back calculated a tick finding rate that would result in that body burden, and then put that into the model.
I
Um,
see.
yeah.
I'd be just curious. I mean, we had a study years and years ago with Sam Telford that didn't find that shrews were as important as reservoirs as mice were.
Uh-huh. Yeah, I think it's tricky to get at it. I mean, I know that some of the Ostfeld papers find that shrews are important, and I think some of the recent work from Telford's lab looking at the where they get the remnant blood meals. Um, in some of those, a lot of those nymphal ticks show remnant shrew blood, but in some of them they don't. So I do think you're right that it's very site specific about where the shrews are the important hosts or not.
Hmm. Okay, thank
But
you.
you're right that the model is the output is gonna be very, very sensitive to, what you the host community that you put in there. And if you don't trust 50 shrews per hectare, then you don't trust the output of the model.
Right. Thanks.
Yeah.
We still have time for another question if anybody has one.
David, this is Kirby Stafford, but you zeroed out the shrews in the model that you actually implemented, correct?
No, I'm sorry. Yeah. So in the model that I implemented, these are the numbers. And so I then tried the model a second time where I zeroed out the shrews to try to tease apart why those bait boxes were not very
Right.
effective in the first one. So in this is with the 50 shrews per hectare. And so that my thought, I was surprised by that. And I was like, why are these bait boxes not very effective? And I thought, oh, maybe it's because of all these shrews. And so then I ran the model a second time where I zeroed out the shrews and then the bait box effectiveness like picks up. So, and this sort of gets to Thomas's question of, you know, if the results are quite sensitive to the number of shrews.
Well, yeah, well, that definitely shows it quite well.
Yeah.